Thanks for inviting me to give you a talk here. It's a very exciting experience for me as well. I think it's one of my first talks in this kind of setting, so I'll try to be as understandable as possible, and if you don't understand something, if I go too much into deep science, just please raise your hand and ask the question. And so, all I have to say, when I told to my grandparents some time ago that I decided to study biology, to become a biologist, and they asked me, so why, who are you gonna be? I said, I'm gonna be a scientist, and I'm gonna study insects. And they said, why? Why don't you want to do something more important, like become a doctor and help people to be healthier? And I didn't really have a very good answer to that, rather than I was just insects are so interesting, and I want to understand how they function, how they adapt to environment and world, and how they cope with stressors. Now, when you put it this way, a few years later, I think actually we reached a place where we need to understand insects very well, because insect health, for first time probably in the history, is really important. Why? Because insects are disappearing. And I'm sure many of you have read news about it, that we are losing our insect biodiversity and the biomass. And while we still kind of dispute which papers are more valid and which methods we should use to account for insect losses, what kind of rings through all these reports is that we are losing 70 to 80% of insects. And that can't be good. Why are insects so important? Well, they're a very important part of the ecosystem. They provide food for birds, for fishes. And of course, maybe many of us are a little bit happy about having not so many mosquitoes on summer nights. Well, I've been doing a lot of field work in Finland, and yes, summer evenings in Värminen can be really, really annoying if you want to be outside and have a barbecue and you're just swarmed over by a horde of mosquitoes. But I have to admit that last 2 summers it hasn't been the case really. We can easily sit outside in June, July and have a beer and a barbecue. There are not so many mosquitoes, the single ones, you don't need to cover yourself with off. So something is really happening. And maybe the ones of you who are having a car, if you drive during the summer on a highway, the windows are not getting that dirty anymore. I don't know, have you noticed it? I've noticed that. Because earlier it used to be, you just drive an hour on a highway and the window is totally covered with dirt. Now it's not the case anymore. So something is happening. And okay, so we have this maybe not so nice insects, but where we should be paying a lot of attention is that next to mosquitoes and horseflies, we are also losing very important insects, namely pollinators. Why are pollinators so important? They are the ones who actually guarantee the fact that we have something to eat. So one would say the wheat is not depending on pollinators and rice is not depending on pollinators. So one of the 2 major crops in the world. But apples, strawberries, pears, oranges, all this high quality food containing a lot of vitamins that are really essential for our well-being are depending on these little helpers. So if they disappear, we will not have apples like we have them right now. We will not have oranges. And actually, the things are getting so bad that in some regions of the world, a lot of fruit trees are already hand-pollinated. And believe it or not, I think that was in Spain or was in Italy, there is actually a startup who offers the services to come to your orchard and help you to hand-pollinate with a Q-tip your orange trees, because there are not enough pollinators around anymore. So what has led to this situation that we are losing them? And just talking about numbers, every fruit bite you are taking on a daily basis is thanks to the pollinators. Coffee, among other things, very important thing in Finland. How could you live without coffee? Well, I can. The coffee yield is up to 30 to 40% depending on pollination. So it's not 100% depending on pollination, but you can imagine if you will lose 40% of coffee yield in the world, how much a cup of coffee can cost. It can become a luxury product, but not so many people can afford. So that's among other one things. So why are they disappearing? Insects and pollinators specifically. Well, one of the things is, well, we are destroying their homes. If this is very schematic view, of course, how the natural habitat, the natural home of a lot of insects and pollinators would look like, then we have turned it into something like that. What for them is basically a desert. There is nothing to eat. There is absolutely nothing. That would be if you would go to Sahara and trying to find the food. This is same for pollinators, so they are just starving there. In addition to not really having much to eat, what we apply in this environment is a lot of toxins. Because in order to grow these crops, of course, there are plenty of insects who would be able to feed on these crops, but we want to get rid of those, so we apply huge amounts of toxins on it. And I'm sure you all have heard about neonicotinoids. It's been widely debated. We're trying to ban them in the European Union and in some other places, because there's been straight correlation between honeybee losses and application of pesticides. So we need to find better ways to manage the crops, more sustainable ways, because among other things, killing the target organisms, unfortunately, neonicotinoids also have effect on pollinator losses. One more thing what we have changed about honeybees is how they live. So this is maybe something how a natural beehive could look like in nature. This is how they probably would like to live if they could decide on their own. What we've done out of it is something like that. So we have absolutely industrialized their lifestyle, right? So instead of living in a forest next to variation of food sources, they are living in apartment buildings what are taken from one corner of the country to another corner of the country, with having very different food. So imagine having to eat 1 month only rice and next month only potatoes. How do you think you would feel? And then you live in a very high densities at the same time. So there's a lot of bees around them. And this kind of mass rearing, well, we know it well, how it looks like. There's a lot of useful animals who live like your chickens or cows or humans. And what is one of the consequences of mass rearing? If you don't have a good nutrition, you have a lot of toxins in the environment, and you live in high densities, of course the diseases will start to spread because your resistance mechanisms are not so good anymore. And the same has been happening in human population for centuries, until there was a breakthrough technology invented in 1796 in medicine by Edward Jenner, who invented the very first smallpox vaccine in UK. And the vaccination is nothing more than controlled exposure to selected antigens to induce specific immunity. And we have learned to use this methodology to prime ourselves against infections. I think all of you have all been vaccinated against one or the other disease, and I'm sure it's been very, very useful. But also we learned to vaccinate our commensal animals. I mean, you cannot actually travel with your cat or a dog unless it's vaccinated. And rabies vaccine is mandatory, I think, for all the dogs. So there is no way you're not even allowed to have animal if it's not vaccinated. And what is special about this feature of our immune system is that you can also transfer it from one generation to another. So as you know, especially in freshly born babies, it's very important that they get protective antibodies from their mother. And these antibodies would defend the babies against the same diseases the mother has been exposed to before. How is that possible? Well, we all have, so all the organisms on Earth, including actually plants, this very sophisticated physiological adaptation called immune system. And it's broadly divided into two parts. Innate immunity, what is a very general immune response, and absolutely every single organism on Earth has it. If you look at the time, then this is so-called first-line defense. As soon as you encounter a pathogen, innate immunity is the one that kicks in. What is it based on? It's based on physical barriers, like your skin, for example. That's a part of innate immunity. Then also your gut lining. If you eat something bad, your gut lining defends the organism so that this infection cannot enter your body. Or the lung tissue, the same thing. And should this pathogen be able to still cross it somehow, there are different cells, like phagocytes and natural killer cells, that are circulating in a blood, attacking these pathogens and actively destroying them. And in addition to that, there are various molecules and proteins that are also able to recognize the pathogens and destroy them. And absolutely all the organisms have this very rapid, very fast, very powerful defense mechanism, including insects. Now, what is special about vertebrates? They also have adaptive part of immunity. What means that later, after the initial immune response have passed, so that is the days later, and the organism has survived, so that's why innate immunity is so important, actually. If you don't have a powerful innate immune system, there's no chance that you will have ever adaptive immune response, because you need to survive this first level. It's an adaptive immunity later. And that means that your organism is later able to recognize the pathogen specifically what you've encountered before. And that is basically the basis for vaccination. That's why we are able to vaccinate ourselves against infections. And for that, there are special cells, B lymphocytes and T lymphocytes. And as a result, we're going to have antibodies that are circulating in the body, able to then recognize pathogens and destroy them. So, like I said, and this has been then basically the basis of vaccination. So if you get vaccinated, what happens is that the special cells in your body recognize the antigen of specific infection, be it the virus, for example, the flu vaccine. Many of us get flu vaccines every year. Immune response recognizes it, produces specific response, and in the end of the day, there will be memory about encountering these pathogens. And then, should you get the actual infection with the flu, let's say 2-3 months later, your immune system already is ready to attack it, and you don't get sick. So, that's what happens in our organisms. Now, bee immunity is somewhat different. And it's very complex, very complicated. It has individual defenses, so that is on the level of a single bee, but can be both behavioral and physiological. And on a behavioral level, for example, bees are secreting special saliva with what entails antimicrobial compounds, and they can lick each other and clean each other with that. Or they also have all these kind of cells and molecules in their blood that are able to recognize pathogens. And then they have colony level immune defenses. So the hygiene has not been invented by humans. The bees are very hygienic organisms as well. So they are able to take out, for example, dead bees from a beehive so that the infections wouldn't spread. They actually also sterilizing their food and their hive. The propolis, what they apply in the hive, has antibacterial properties. So there's also colony level immune responses there. And also what is known that sick bees quite often leave the hive not to infect the others. But what they don't have is antibodies. So and researchers have been looking for antibodies in bees, but they've not found any. And hence it's thought that bees don't have any ways to recognize once encountered pathogens later. And always when they encounter pathogen, they need to upregulate all the immune responses in one go. Hence vaccination in bee world would not be feasible nor possible. At the same time, bees are having a lot of threats. There are a lot of diseases, there are a lot of viruses who attack bees. Among one of the most important ones probably is the deformed wing virus, you've all heard about it. I think mostly we hear about the mites, varroa mite, but actually why varroa mite is so bad, it's transferring the virus. You could almost say it's a malaria and mosquito story. So the mosquitoes themselves are not so bad, but the malaria they transfer is bad. So here's kind of in a way similar situation where varroa is transferring the virus, deforming virus, what will infect the bees and will cause different things, and that's only. Then there are a lot of fungi that attack bees, but also a lot of bacteria. Among others, a lot of bacteria what are really infant diseases. Why they are so dangerous is because they are attacking freshly born babies. So the adult bees would be resistant against the disease, but the babies are not. Like American foulbrood, also common in Finland, especially in southern part of Finland. And yeah, so what happens that babies die in the hive and hive dies eventually. So, like I said, so what do we do when the bees get sick right now? Well, actually, to be honest, the number one advice is to burn your beehive. Number two advice is to apply antibiotics, because there is no way to get them healthy or vaccinate them against diseases. So antibiotics are discouraged in many areas, including Finland, because one penalty you get is you cannot sell your honey later, because nobody wants to buy honey where are antibiotics inside. Or you could not sell wax, what is used for medical purposes or for cosmetics. So antibiotics, not so good. And also the diseases are getting resistant against it. So in the end you would have to apply more and more and more. So that's what you do if you have a diseased beehive. You close it in the evening when all the bees are inside, pour it over with gasoline and put a fire on it. As you can imagine, that's not a nice thing to do. Nor really very sustainable in the long run. I mean, you're losing your production animals and kind of your pets, right? It's a living organism. So we obviously need alternatives to that. And to do that, we need to be able to better understand immunity in insects and in bees. Now let's take a side step. Like I said before, no antibodies in insects, hence the immunological memory should be impossible. And especially also transfer from one generation to another. Now, in the beginning of 2000s, what researchers started to notice that, it's not that simple. And there seems to be transfer of a knowledge of encountering pathogens from one generation to another, also in insects. And that in bumblebees, in honeybees, in moths, but also in water fleas. So it's not that simple that though they don't have antibodies, somehow they still are able to transfer the knowledge of encountering pathogens. And being then basically, as a result, the babies are more resistant against infections. So the central question here, of course, is what is the mechanism for it? So how is that possible without antibodies? And here I'll take a side step, and that is something what I did freshly after my PhD. When I got really curious about this phenomenon. So how does that happen on a physiological level? So what is the mechanism? And I worked here on a wax moth, so not too far from honeybees. As you know, wax moth is a brood parasite of honeybees. Smells very good, the food you feed wax moth. By the way, it feels like a muesli. So if you grow them in a lab, it's really, you open the boxes where there's wax moths inside. It smells like a muesli and bread. So where's what I was thinking? So how do we start tackling this question? So bacterial infection. How do you get infected with bacteria? If you get salmonella, how does that happen? You ate something bad, right? So, and that's actually how most of bacterial infections are encountered by the organism. So you eat it in. So my question was, so what happens to this bacteria actually when the organism eats on it? Can we somehow follow them? So can we tag them and see what happens after, this wax moth larvae has been eating bacteria? Can I just see what is happening to them? And I don't know if you can see it, but you have to just trust me. So I use tagged bacteria, so there's like a mark on this bacteria, so I could recognize them. And what I could see that you can detect them that they are in the gut, so they are definitely in the food. Then you can see that they actually go to the body cavity, so they are inside of the organism. And you can see, so these green dots, these are labeled bacteria. And they're also taken up in the fat body. So fat body is a special organ in insects, somewhat similar maybe to human liver. But very important, there are a lot of things are being stored there and a lot of proteins are being produced in fat body. Among others, also a lot of immune response related proteins. And it seems that these bacteria, so they get digested, pieces of them are transferred to the organism and they aggregated in a fat body. What happens next is a question. So does it cause something? So if I follow now a female wax moth, what I saw that these bacteria actually transferred to the reproductive organs. So now I said, why would they do that? So of course my question, so actually are they passing these little pieces of bacteria from one generation to another? So if we see them in a reproductive tract of females, could we also see them in the eggs that females lay? And yes, we can. So in a way, we can track the bacteria, or actually pieces of bacteria, from the food to the eggs that female is laying. Why is it so important? What are these pieces of bacteria? Now let's ask, what are the vaccines? Vaccines are nothing more than pieces of bacteria. So what we do when we create whole cell vaccines, we take bacteria, we kill them, fragmentate them, and then we inject them. Because the immune system is able to recognize these little pieces. And it seems that insects are transferring those little pieces also from one generation to another. And now if we look at the immune responses of these eggs who've got these little pieces of bacteria, we see indeed that a lot of immune response related genes and proteins are upregulated. Basically it means they are ready to tackle the disease should it come. Still, this does not answer a question, how do they do that? So somehow they transfer, if you like, this vaccine from one generation to another. And that was a time when I came to Helsinki for my next job. And I met a wonderful lady, Heli, now Salmela, who was working on this wonderful protein called Vitellogenin. This is a very fancy 3D structure of it. This is how we see it. But actually, what you should think when you think about Vitellogenin is the egg yolk. And depending on the animal, up to 98% of the egg yolk is consistent of this protein. So it's a really important protein. It creates a basis for nutrition for a developing embryo, for example. So it's really essential. And it's present in all the egg-laying animals. Whether it's a turtle or a chicken or a snake or a fish or a honeybee. So a story what Heli was telling me when we met that in fishes it's been showing that Vitellogenin is able to bind to bacteria. And I was like, hey, other Vitellogenin is synthesized in a fat body. Remember the cream glowy tissue in a wax moth. And I was like, hey, let's make a bet. Vitellogenin in honeybees will take the pieces of bacteria to the eggs. I said, okay, let's give it a try. So what did we do? We ordered a few honeybee queens in a lab in Helsinki. We dissected them and they have paired ovaries, right? So now we used the same labeled bacteria what I used also in a wax moth. And we incubated part of the ovary with labeled bacteria without Vitellogenin and part of it with Vitellogenin and labeled bacteria. And yes, indeed, what we've shown there, that the mechanism how this immunological signal gets from one generation to another is linked to Vitellogenin. So you have this red glow here in the eggs of the honeybees, shown with confocal microscopy. And remember, these were the eggs and wax moth. So we see basically a very similar picture here. Now we just know what is a protein responsible for it. So with that, we came up with a hypothesis that this is how it happens. We assume that when the honeybees go out and forage, they pick up the pathogens from environment, bring it back to the hive, where it's fed to the queen, bacteria are broken apart, and then Vitellogenin, so this egg yolk protein in a queen, takes these pieces of bacteria to the developing eggs, where this can prime the next generation, and all these little eggs she's laying, which you can see there, all the larvae that come out from there are more resistant against infections. Yeah?
Sorry to interrupt you, just to check. So the bees that catch the bacteria and bring it to the queen, they might die for it because they don't have the immune system for it, but then the next generation after the queen eats the bacteria would be resistant.
That would depend on infection. Now, if we talk about American foul brood, what has been our model right now, then the adult bees are resistant against it. So that is the tricky part about this infection. So the adult, they are absolutely not sensitive, so bacteria is not able to infect them. The only ones who are actually susceptible are freshly born babies. The babies who are up to 48 hours old. After that, they become resistant. So this is really very crucial that this very first life stage would be more protected. So that's why actually the adult bees can bring it back to the hive. They don't suffer from it at all, but the baby bees will suffer. So that's what it also makes this disease very hard to combat. So even if you would throw a lot of antibiotics on a hive, in reality, the very first bite what the bee baby takes could be the last one. If there are bacteria inside, it could kill it. So that's why anything what mother could do to make the babies more resistant would be worth a lot. So, but of course, now comes the tricky part if you know bees. And so the experiment we did was in a lab with a bee, his histology dissected, nothing really natural if you like. So how does a beehive look like? There are two different types of individuals in a beehive. There are sexuals and workers. So sexuals are the ones who produce brood, like a queen. There are also drones, males, but they're not in not in a beehive. And then we have worker bees. So we have nurses and foragers. And queen and nurses, they do not leave the nest actually ever. So they would not have direct contact with any environmental bacteria. So it doesn't matter what it is. So they don't really encounter that. So how does queen get the signal about that there might be some infection around? Because who leave the nest are the foragers. They go out and bring back the food, the nectar and pollen to feed the colony. And then nurses take this and feed the queen and the brood. So how does that fit together with our model? So what we did then was to use the nurse bees who get the food from the foragers, right? And we fed them again with the same labeled bacteria to trace, so how do they get this signal from their food? And what we showed is that, again, these labeled bacteria, so these are these red little fuzzy things, they go through the gut, like it was in a wax moth, so they are taken up by the body. You can see them in a fat body, again, so they are accumulated in a fat body. And most importantly, they also transfer to the head glands of the honeybee workers. Why are head glands so important? Because head glands, so this here, is a place where the food, the jelly, worker jelly and royal jelly is produced for a queen bee and for a larvae and secreted and fed to other bees. And we showed that actually this immunological signal is seen in this jelly, what they produce for a hive to feed on. So basically, we could say they pass the vaccine on. Now, this is all nice. This is all in a lab, labeled bacteria. One could ask, does it actually help against real diseases? And that is experiment We did in Helsinki, in Viki. And we used 2 different pathogens to prime our bees against this American foul brood, it's a bacterial disease, baby disease, and child brood, that's a fungal disease. Again, infects larvae in the first line. We had these little beehives, the so-called mating hives in the Viki greenhouse area, maybe some of you have seen it. And as a result, not going into too many details, but yes, it does. So we've shown that in both cases, against the American foul brood and against the child brood, if the queens get priming treatment, the larvae are more resistant against the diseases. So they too pass the immunological signal on from one generation to another. And with that, we hope that we've opened maybe a new frontier for bee health. And maybe we can save a little bit of a world with that, because bees are important, and without them I don't think our French table will be as tasty as it is today. And last but not least, I would like to thank a lot of institutions, a lot of people who worked with me on this wonderful project. And thank you for listening.